Hello everybody. So, let us discuss about the module 3 in the part of our course. So, first of all we will discuss about the osmolytes. I have mentioned earlier the osmolytes or osmoprotectants are synthesized in our plant system upon exposure to different stresses, and those osmolytes protect different cellular organelles, different proteins from the harsh exposure to salt stress or the harsh exposure of dehydration. Okay. So, in natural system in our cell, some amino acids like cysteine, arginine, methionine, it constitute around 50% of the total free amino acids, and once the plant encounter the salinity stress, then their level of cysteine, arginine, methionine is drastically reduced, and at that particular condition the proline concentration is raises in the system, and proline is a very important amino acid. Okay. So, the intracellular proline, it does not only provide the tolerance towards stress, but it serves as an organic nitrogen reserve during stress recovery. So, during stress it will protect some of the proteins or some of the organelles, and once the stress is relieved, thereafter at that stage also it will help the plant. Similarly, another important osmolyte is glycine betaine. It is a non-toxic cellular osmolyte, and it will improve the osmolarity of the cell during stress period to mitigate the stress, almost similar way as the proline works. So, glycine betaine also protects the cell by stabilizing proteins and protecting different photosynthetic apparatus from the stress damages, as well as from the different detrimental effect by the reactive oxygen species like superoxides, hydroxyl radicals, etc. So, let us discuss about the biosynthetic pathway of proline amino acid, which is mostly expressed or which expression is increased upon exposure to any type of stress, and here we are discussing its biosynthetic pathway in chrysanthemum. So, from two different sources, either from glutamate or ornithine, it is produced. So, from glutamate, by the action of pyrroline 5 carboxylate synthetase, the glutamic gamma semialdehyde is produced, whereas from ornithine, by the action of this enzyme, the ornithine aminotransferase, the glutamic gamma semialdehyde is produced. So, once glutamic gamma semialdehyde is produced in the system, so it can produce the delta 1 pyrroline 5 carboxylate. This is the immediate precursor of proline synthesis. Once the pyrroline 5 carboxylate reductase enzyme will work on this substrate, the proline is produced. Alternatively, another enzyme known as proline dehydrogenase. Okay. So, this enzyme basically degrades the proline, and from proline it will produce delta 1 pyrroline 5 carboxylate. So, in the system in chrysanthemum species, upon exposure to salt stress, it was found that those three genes highlighted as yellow, that is this one, the proline 5 carboxylate synthetase, the ornithine aminotransferase, and pyrroline 5 carboxylate reductase, their expression is upregulated upon exposure to salt stress. So, it means if their expression is upregulated, means finally the proline production will be increased, and at the same time the expression of proline dehydrogenase is down regulated in salt stress. It means the plant try to accumulate more amount of proline under salt stress by over expressing these three enzymes – and by down regulating this particular enzyme. So, in this way different approaches are taken by different scientists to do the metabolic engineering to finally get the stress tolerant mechanisms. So, several scientists have worked on genetic engineering aspect. So, genetic engineering aspect of plant we will discuss about these things hence onward. So, through genetic engineering we can modify the genomic architecture or genetic architecture of a plant by transferring a foreign gene into its system. Earlier different direct gene transfer method was available like bioelastic gun, gene gun or liposome mediated transfer, but most of the favorable transformation process is done through direct gene transfer by application of Agrobacterium tumefaciens strain. It is a bacteria which can transfer the part of its genome into the host genome, into the plant genome. So, by Agrobacterium mediated several genes have been transferred into the plant system by different scientists, and finally we got several abiotic stress tolerance like heat tolerance, herbicide tolerance, salt tolerance, heavy metal tolerance and drought tolerance in plants. In US most of the soybean which is grown over there, those are herbicide tolerant, and several genetic engineering has been done in their genome, so that in a crop where those particular soybean varieties are grown, then we can apply herbicides, the weeds will be killed, but the plants, the transgenic plants, genetically engineered plants will not be killed, and it will help in mechanization. The labor cost will be minimized by those type of transgenic development. Then through several transgenic developments we got the fungi, bacteria, virus or nematode resistance. Since earlier time for viral resistance, in most of the times scientists overexpress the coat protein genes in the plant system, because we know in the virus the coat proteins are available. So, once those proteins are expressed in the transgenic plant, if the virus infect the plant, then the viral DNA or viral RNA will be encapsulated within those coat proteins. So, the viral infection is not visible. So, in this way several transgenic plants have been developed which are resistant to virus. In case of fungi, bacteria as well as a nematode, several pathogenesis related genes or germin like protein genes or germin genes have been overexpressed. To control several biotic and abiotic stresses in together, a lot of studies have been done on overexpression of transcription factor genes, because transcription factors plays as a master switch. If we overexpress those transcription factors, at the same time we can control several abiotic stresses. Okay. In addition to that, genetic engineering has also been done for quality improvement in crops, like in case of rice, the golden rice have been generated to improve the vitamin A content. Then several genetic engineering has been done for changes in architecture. Certain changes have been done by changing the root architecture of the plant by overexpressing or down regulating certain genes through genetic engineering. And once we can overexpress or down regulate a gene, and if a plant changes its root architecture, then it can show either drought or salt resistance mechanism. Okay. Because in most of the things, if the root structure is become improved, if the root structure becomes more robust, the plant can tolerate the drought stress or salt stress in a better way. So, certain studies have been done to overexpress the transcription factors or osmolytes, okay, as those are the master genes. So, later on we will discuss those things little bit in detail. So, let us discuss that the how transgenic plants are developed by the scientists. So, first of all we have to prepare the gene construct. So, initially the gene construct is prepared in smaller vector like puck based vector, and thereafter it is transferred into binary vector. Okay. So, for simplicity I have just made the diagram in such a way that a vector is available. Vector is in most of the cases for molecular cloning we do a circular plasmid vector, okay. And inside of this vector if we have to express a gene we need at least 3 different things. One is the promoter. If a gene does not have a promoter it will not be expressed. So, we need a promoter because in promoter RNA polymerase will bind and it will express the gene. Then we need a gene sequence where from ultimately we can get our desirable protein, and third one is the terminator sequence or transcriptional terminator sequence where the transcription will be stopped from our targeted gene. So, these 3 things are basically gradually cloned one by one or at the same time inside of a vector. So, once it is cloned inside of a vector the gene construct is initially ready for its further process. So, first a gene construct is made, then the binary vector containing this gene construct is transferred into Agrobacterium tumefaciens. Agrobacterium tumefaciens is a soil bacteria. It has the ability to transfer certain part of its tDNA or transferred DNA into the plant genome. So, if we place our gene construct inside that tDNA, then by the help of this Agrobacterium tumefaciens our tDNA will be transferred into the plant genome. This is a natural mechanism of this bacteria, and a particular research depicted that a lot of sweet potato varieties are available in the market in which the Agrobacterium tDNA is integrated in the genome of those sweet potato, and we are eating it, we are having it and we are not facing any problem. So, this is not harmful. So, then if we can make this type of construct and if we deliver this construct into Agrobacterium tumefaciens strain, so thereafter we have to transfer this Agrobacterium harboring this particular plasmid into the plant genome. So, for that reason we need to do plant transformation. Okay. Now, in this system, in this particular gene construct several selectable marker genes are available. Some selectable marker genes are available for bacterial selection, because if after transforming it into Agrobacterium we are getting a large number of colonies, but our desirable plasmid may not enter in every colony. So, some selection is needed. Some antibiotic resistant genes are used as a bacterial selectable marker, okay, like kanamycin, rifampicin, chloramphenicol, those genes are used as bacterial selectable marker. So, based on that we can screen that particular Agrobacterium colony where our desirable construct is available. So, once we will have our Agrobacterium strain with the desirable construct, we will infect this to our plant material. So, our plant material in case of rice, the rice calli is used for plant transformation. In case of tobacco plant, the tobacco leaf disc is used for plant transformation. So, either the callus or the leaf disc is suspended with the Agrobacterium solution, and that Agrobacterium is harboring our particular gene construct. So, once the plant part, the callus or the leaf disc is transformed with the Agrobacterium, then it is placed in selection media. After co-cultivation and those things, it is placed in selection media. Now, another selection is needed. Here we have to use a selection gene or selectable marker gene, which is for plant selection. In most of the biotechnological research, the hygromycin gene or kanamycin gene used for plant selectable marker gene. Okay. So, after transformation, we may see a number of seedlings coming from the rice calli, or a number of seedlings are coming from the tobacco leaf disc, but among them all of the seedlings may not have our target gene. So, if the Agrobacterium tDNA contain the selectable marker gene specific to a plant, which is kanamycin selection marker, kanamycin resistant gene or hygromycin resistant gene, then the plants which will acquire those plasmids, it can be grown in kanamycin media. So, rest of the plants which does not have the kanamycin resistant gene will be killed, but the plants which have the resistant gene, which is coming from our construct, will be gradually grown and survive. So, in this way we can get the putative transformants, and thereafter we have to do the subsequent analysis. We have to confirm the transgenic plants by southern hybridization, by northern hybridization, by RT-PCR, by qRT-PCR, and we can do different stress tolerance study. If we are screening for any abiotic stress, if we are expressing any transcription factors, then after getting the putative transformants, we can do the subsequent study. Now, these are the different transgenic approaches which have been taken to mitigate the stresses by several scientists. First of one is the overexpression of late embryogenesis abundant gene. It is a LEA gene. It is very important gene, and it was found to show the drought tolerance in rice, not only in the greenhouse condition, in the field condition also, in natural condition also it showed the drought tolerance mechanisms. So, from the last slide it is clear that how the LEA gene construct is made. The gene should be placed after some promoter, and after the gene some terminator sequence should be there, so that the gene will be expressed, it can produce its RNA and protein inside the plant system, and by the action of this particular protein finally we can see its phenotype, we can see some tolerance mechanisms. Then another study, there the Wes DREB gene, DREB, that is dehydration response element binding proteins, binding factors. So, this group of genes which is responsible for drought tolerance or abscisic acid mediated different signalings. So, by Chen et al. In 2008 they have expressed the Wes DREB gene in rice, and they also found the improved drought tolerance mechanisms. In another study by Ghonti et al. In 2011, they have overexpressed P5CS gene. What is P5CS? It is a gene involved in proline biosynthesis. Here you can see that P5CS, that is pyrroline 5 carboxylate synthetase, that is the first enzyme which converts glutamate into glutamic gamma semialdehyde. So, this group of scientists, the Ghonti et al., they have basically overexpressed this particular gene in chickpea, and they have found the salt tolerance. So, once they express this P5CS genes, they found that the amount of proline have been increased in the plant system. So, proline which is not which does not only maintain the balance, does not only protect different organelles or different proteins under stress situation, it will help during stress recovery also. So, the proline accumulation upon overexpression of this particular gene increased a lot, and so finally we can see a lot of stress tolerance. Then in another study they have overexpressed a TA MYB33 genes. MYB is a MYB group of transcription factors from TA, that is Triticum aestivum, from which this gene was cloned, Triticum aestivum, and it was overexpressed in Arabidopsis system, and it was found to show improved salt as well as drought tolerance. So, it was reported by Quinn et al. In 2012. So, these are the reference of this particular course of today's topic. First one has been published in Plant Cell by Liu et al. Then Xiao et al. It was published in Theoretical and Applied Genetics. Then Chen et al. Published their work in Biotechnological Letters. Then Hubbard et al. They published their work in Genes Development. Then another study by Quinn et al. It was reported in Molecular Biology reports. Then Denlin et al. It was reported in Trends in Plant Science, and Ghonti et al. It was reported in Biologia Plantedam. So, that is all for today's lecture. Thank you all.